President Lee and Professor Byung-Lee Lee and Professor Choi, and to members of the National Academy of Science. I wish to thank you for the honor of having been elected honorary member of your prestigious academy. It is humbling to be selected by this august body of respected and successful scientists. Thank you again. I also wish to thank Professor Byung-Lee Lee and Professor Choi and others of the fifth committee who organized this symposium for the invitation to speak in this opening session of the 48th International Symposium put on by your academy. I do so with pleasure and hope that my remarks are taken in the context in which they are intended. And thank you, Professor Choi, for your kind introduction. I wish I were there in person to meet you and other members of the Academy. That was the original plan in 2019. It has been a number of years since I visited your lovely country and have fond memories thereof. I have had the privilege of hosting several postdoctoral fellows from Korea in past years and valued their input to my research team through my many years as an active research scientist. Now, in this talk, I am taking many liberties. My intention is to stir our thinking as scientists and influencers in the academic setting in which many of us work. Much of the information about my past is shared in the biographical sketch that you may have read. You may also have read the brief paper that is printed in the proceedings book. If you have read it, you will know that my talk will not be of science per se, but of the role that we as scientists play outside of the laboratory. Yet our laboratory and our classroom activities lead and focus our roles outside of our institutions. We are influenced by many things, and our experiences are individual. They are unique from the experiences of other scientists. Yet, for all of our differences, our goal is to serve society, either through discoveries, applications, or influences and activities that come through policymaking. This is the spirit in which I present these remarks. And I welcome feedback in whatever way you may wish to engage. For example, through the email address that is shown at the bottom of this first slide. So if you will permit me, I will proceed with a few remarks. And first, I want to tell you, share with you my obligations and current commitments in an effort to be transparent in my remarks. As a professor emeritus at Washington University, I have few responsibilities. However, I serve on the U.S. National Academy of Science and the National Research Council on the Committee on Policy and Global Affairs, and recently am a sub-chair of a subcommittee on U.S. and China science diplomacy. This is a new activity through which we are encouraging U.S. scientists and Chinese scientists to engage in those areas that we hope will be of common interest and will help contribute to peace between our nations. I also serve on the National Science Board of the National Science Foundation. I serve on a subcommittee from the White House Office of Science and Technology Policy, the subcommittee on biomanufacturing. I'm a consultant in private industry with a company in Ireland called Talum Biotech. It's a microbiome company. Performance Plants in Canada. It's a genetics and GM genetic modification company. And Cultivate, a company in St. Louis, Missouri, my hometown, where we are working on a new project on biomaterials. I have served for many years as a Council of Science Advisors at the International Center for Genetic Engineering and Biotechnology, which is headquartered in Trieste, Italy, with laboratories in New Delhi and in Cape Town. This range of activities provides me an opportunity to look at science from a different perspective, not the perspective of a hands-on day-to-day scientist at this stage in my career, but rather to provide advice and input where it is requested, with the hope that it will make some effect and have a difference in our national programs and our international activities. Now, the National Science Board has two roles, and I want to only highlight this institution for which I serve, on which I serve. It's the policymaking body for the National Science Foundation. It relies on comments that it makes that are critical to NSF's future, for improving strategic budget directions and major programs and awards. But its major function outside of the National Science Foundation is to be an advisor to the President and the Congress. We publish a biannual report on science and engineering indicators, a very data-rich compendium of the activity of science around the world, not only in the US. Korea, of course, South Korea is, of course, mentioned in that report. Issues and policies reports on science and engineering, on STEM education, and on the workforce development, an important component which your president mentioned and in his remarks. As part of that activity, I was a chair of a committee that developed the Vision 2030 for the National Science Foundation and for the board, helped to direct our activities. And we summarize there in that report something that you have in Korea also faced. How do we ensure as scientists and policymakers the success of the R&D enterprise in our countries? The science and engineering ecosystem is a combination of activities that span the academies, whether it's trade schools or universities or the K through 12 education, the early education. We have engagement of the government at the state, local, federal, and in national laboratories that are sponsored by government activities, and thus the taxpayers' money. So we are responsible because we are funded through government funds. There's also a heavy engagement by the private sector. In the US, there was more research conducted by the private industry these days than by government sponsorship. But there are many businesses and associations. It involves entrepreneurs. We have many public foundations in the US and nonprofits and philanthropies. These all work together to ensure the success of the enterprise writ large across many areas of science. We know that, as was mentioned in the opening remarks by Dr. Lee and Professor Lee and the president and others, that science and technology together will improve the human condition in the face of a changing world. I consider myself a realistic optimist, but I'm also at this stage in my life more realistic about what we can achieve in the short term and the medium term and the long term. We know that science has the capacity to deliver the knowledge. Technology has the potential to deliver the benefits to the consumer or to the public if commitment to societal goals is embraced, and embraced across the full circle of the success. There's a requirement of our science and all that we do to achieve a sustained science-driven future, which must include essentially recognizing the challenges and setting a pathway to specific goals. Those goals are important for us as scientists and for those who sponsor the research, etc. Those goals must be agreed by scientists and consumers, citizens and consumers, governments and the private sector, and to engage the scientific leadership and those who are the directors of science. To do that, of course, we must ensure citizen input and buy-in. We have suffered in the recent past for the advances of science that are then rejected by a public because of the way the priorities were set, or the goals of those who would deliver different messages. We need also to provide freedom and resources to scientists to explore and discover solutions. However, it's increasingly important that those goals be guided by established frameworks that are set either by the scientists themselves, or by industry, or by consumers, etc. Going along in parallel with the discoveries is the developing of science-based regulatory frameworks that guide prompt and timely delivery of new and novel products from science and technology that make their way through the technology transfer mechanism to industry. And we need an innovative private sector that embraces new products and delivers those products at affordable and sustainable cost. Of course, the private sector also requires sustainable profits. There are much evidence that science and technology delivers the goods through discovery, collaboration, and application. These are simply several of them, but I want to highlight the importance of discovery and collaboration, because in this world of complex challenges, complex goals, we need discovery and collaboration to go along with application. Collaboration across areas that are not comfortable to those of us who are individually specialists, perhaps as physicists or as mathematicians or as geneticists or genomic scientists. We know from needs-based observations and discovery from the late 18th century onwards, there are many examples that indicate that science can deliver the goods. Early examples of vaccines and antibiotics, clean water, improved agriculture that led to safer foods and extended a lifespan as a consequence of healthy agriculture. In the 14th and 15th centuries and 16th centuries, we had much less safe food and lifespan was short. We've had through this work improved transportation, better roads, and so forth. Recent examples include many breakthroughs. Much of my work has been not devoted to a discipline, but rather I embrace a variety of sciences to solve the problems that I set out to solve. But those breakthroughs in cellular biology, structural biology, and biochemistry often lead to applications. New crops and new food technologies, new types of vaccines, cell-based and stem cell-based therapies, medical devices, etc. These breakthroughs happen on a regular basis these days. We hear wonderful stories that come to the lay press as well as to the scientific press. Many of these breakthroughs, however, require collaborations across disciplines. And these disciplines that help to enable applications and that lead to breakthroughs for society. It created new materials and devices, including biomaterials, solutions for renewable energy, detection devices, sensors in all fields, advances in climate sciences and predictability, transportation, whether it's electric vehicles or improved mass transit and so forth. And we've seen applications in math and physics that some of us didn't think would happen, but they did. Not only has it taught us about astrophysics and the space that we are part of, but it's led to new communication devices, new generations of computers, laser-based therapies, and solutions in engineering, and etc. So we know it can deliver the goods. So my own experiences in science and technology comes from a background that was not specific, but very broad. As an undergraduate, I did research of my choosing. And I underlined of my own choosing because that was important for my staying engaged as a young scientist in the field of science. And it's a lesson I think that we need to transmit to our graduate students and to our undergraduates, that there's much to be discovered, but there's much to be applied. I received classical training, and then, I had the freedom to choose a study that I wanted. studies in virology. And at that point, we were doing groundbreaking work in Professor Harry Murakishi's laboratory on cell cultures, at the same time that many of the labs in Japan were working on protoplasts. We discovered and studied in my own work, a cell-based resistance reaction in cell culture, using what was then a state-of-the-art analytical technologies. Young scientists need to be encouraged to explore new technologies. New science comes from the latest of technological advances. Again, the postdoctoral research was of my choosing. Molecular aspects back in the 80s was very different than molecular aspects today, but nevertheless was important in what I did. I then began a second career in cell and developmental biology. That led me to more biochemistry and more understanding of how cells work. And then I combined those two disciplines. And as a professor and a scientist, I conducted research of my choosing with funding from multiple agencies. I had the privilege that is not so easy to come by in many countries. I had the privilege of low classroom teaching requirements so I could focus on my research. These institutions hosted our projects through the years. What made it work for me was excellent facilities and an institutional commitment to excellence in research. And the second part was an outstanding scientific colleagues from wide ranges of scientific disciplines. I had the pleasure of knowing one of the chemists who was awarded the Nobel Prize in Chemistry this week, Dr. Barry Sharpless at the Scripps Research Institute. So having the exposure gives you the new experience that allows you to new things, to push in areas that you didn't push before, because as I mentioned earlier, collaboration is essential to the solution of complex problems. We all study systems, and those systems are complex. The solutions are not mine or yours, they are a group activity. So I want to remind you of some work that we did many years back. We discovered a mechanism for genetic engineering, and this discovery was part of what led me into new sciences. I happened to be at a location where plant genetic engineering was invented at Washington University in St. Louis, which is the same city where Monsanto Company was before they became Bayer. And the collaborations which were possible led me to do this work. This is an infection of a plant with tomato or a tobacco mosaic virus. And we engineered a gene which encoded the coat protein of the virus into the plant and then developed virus resistance as an outcome. That was the first example of a disease resistance in plants, but it's the first of many that other collaborators around the world using a similar technology applied to other diseases. I was very young back then. I don't look the same now. I guess it's consistent with the not only the aging of the science and the changing of the science, but the changing of the scientist as well. The first applications were to prevent the infection of papaya. This may be common in South Korea. It certainly is in Japan and the Philippines, Hawaii, and many other countries where papaya is grown. The papaya ring spot virus is was decimating the production of papaya in Hawaii. Dennis Gonsalves and his colleagues used a technology similar to what our work in tomato used and developed virus resistance. This is a picture of the disease on the papaya that you may recognize, the rings, therefore the name papaya ring spot virus, caused by a simple RNA virus that when engineered appropriately leads to disease-resistant plants. This disease resistance has been stable now for more than 20 years. This is an example in squash, a similar technology, and in cucumber. And so it's important to recognize that the technologies that we develop in our laboratories may be of scientific and novelty, they may be interesting, and they may get us awards, but unless they lead to applications for society, our work is symbolic, not actual. But at the time I was doing this work, there was also a very large outcry against the planting of GMOs for the consumers. And so as a scientist, I changed my field. I changed into mechanisms to understand more of the scientific basis for this resistance reaction. I did so as a structural biologist. Now, I was not trained as a structural biologist, but because the virus that I studied had been crystallized and resolved to a very high resolution, we knew about the structure. We used our knowledge of the structure to change some of the amino acids in that coat protein by modifying specific residues that are that are the contact points between two different subunits, as shown here. We actually made mutations in many different amino acids. And you can see some of those positions here. We then transferred those to transgenic plants and asked which of those would confer resistance. And the result is that some of the mutations just changed the resistance, and the plant became sick very fast. Other mutations led to essentially immunity, a very high level of protection. Now, as a biochemist and a biologist, I find this very interesting and intriguing because it means that the first observation can lead to improvements and second observations. That knowledge of crystal structure was terrible. It was very, very important for us to understand how the virus does its work and how the engineering of plants can be improved. I then had the opportunity to apply a similar technology to a disease in South Asia. This is a picture. I think it may be coming slowly to you. I'll wait for a second. A disease that is of groundnut or peanut, which you see here. These diseased plants are infected with a peanut virus, and we isolated the gene for the capsid protein or coat protein and developed virus-resistant plants. At this point, we thought that we had done a good thing for society because this disease affects more than 10 million hectares in India. And we thought that we would help to solve the problem, but it was not to be. Because of the regulatory impediments and the regulatory hurdles in India, the solution to this disease was never deployed. Today, farmers still struggle to continue to control this disease. That led my interest to trying to understand more about why technology, which is useful, is rejected. What are the things that you do or that I do that could help the technology transfer and adoption by the consumers? Many thought that genetic modification was interfering with the natural order of biology. Plant breeding and genetic engineering are really partners in crop improvement, and they will continue to improve crop traits. The top line shows some effects that conventional plant breeding might use. There's the technology to introduce new genes into the tomato, as we did with our research. There is the use of genome editing to accommodate new changes. This is seen as less invasive. But in fact, all of these have value in the context of our achievement of goals. And our goals in this case are to improve crop yield, to improve food quality and food nutrition, and to increase the livelihood, the economics, the profitability of agriculture. So we see we see genetic engineering and genetic editing as a continuum, one that leads to crop improvement. The question is, how do we get how can we assure that the technology will be accepted? There's recent research which shows advances in many areas, and it gives optimism to the scientists, but it should give us optimism for the future of the agriculture and food sector. In the case of production, for example, improved photosynthesis and nitrogen capture is underway. The research advances are phenomenal, and I'll show you a recent article. Disease resistance improvement, adaptation to changing climates and environments. The company with which I am working in Canada has some outstanding technology for drought tolerance in crops, for root growth, and for other features. It's not the only laboratory. Your own laboratories in Korea have similar advances, but it's toward production of agriculture. There are great advances towards food safety, which are required. The company that I spoke about earlier, the microbiome company, has focused on reducing heavy metal content in crops such as rice and potato and others. It's possible to reduce the amount of phytotoxins and mycotoxins that are in foods to improve the safety of food. Good advances towards improved nutrition, increased vitamin content, healthier oils, improved fiber content, and etc. And of course, there's a great energy and a great demand to work towards environmental and economic sustainability. Agriculture has some problems to overcome in terms of sustainable activity, yet encouraging the economics of the agriculture sector. This should integrating reduced inputs, enhancing microbes, and I was very pleased to see that there's a talk later in the symposium, I think by Professor Young-Ron Chung, on this topic of the use of microbes in improving soil health and in disease resistance. The use of sensors is having an enormous effect on how agriculture is done. So there's a lot of reason to be optimistic, but there are some cautionary notes as well. We called earlier for placing higher priority on developing nutrition-rich foods. This is manioc or cassava, which has been engineered not to be orange instead of white by increasing the beta carotene content. It's not as bright as carrot, but you can see that it's cassava of this type now has higher levels of beta carotene. This product is now in final stages of approval in Nigeria and in parts of West Africa and East Africa, where cassava is as an important part of the diet. There's a technology in potato, which would reduce the browning of potato by using a genetic engineering and a gene editing technology. Papaya, we spoke about earlier. And of course, brinjal, or eggplant, as we say in the US, with resistance to diseases to improve the nutrition content and the quality of the fruit. And of course, there in Southeast Asia, you are well aware of the development of golden rice. And the recent announcement from the Philippines that the golden rice has been used now in commercial sale for the first time and is expected to expand. Very, very high amount of beta carotene in that rice. Transgenic potatoes with resistance to a very important potato disease, potato blight. The harvested tubers are free of the disease, and the farmer no longer requires the use of pesticides. There's some really exciting new information that some of you may recently have seen in the issue of Nature Food in June of this year, a paper which described the inorganic and a biological artificial photosynthesis system for energy efficient food production. If you have not seen this paper, you might want to take a look at it. It's really exciting to think about the transfer of CO2 to acetate, and to use the acetate in a system to feed the plant that is now something in the range of 20 times more efficient than photosynthesis. And it uses all of the carbon comes from carbon capture, a way to capture the carbon dioxide in the air. A recent, again, in food, Nature Food in June of this year. So what limits the adoption and application of advanced S&T? Well, certainly acceptance and confirmation, acceptance by the consumer, and importantly, confirmation by other scientists and by other technologists. Too often, scientists are skeptical of other advances in those fields of which they are not familiar. And having the imprimatur, the approval by other scientists of a technology is very important. You as a scientist have the right and the expectation that you can read other articles, but you should, and know other science. But whether or not I, for example, would comment about vaccines or antibiotics in humans, it's clearly not my field. However, I'm interested therein. We need also to engage a greater amount of innovation and entrepreneurship in academics. This is critical in order if we are to take science from the universities into entrepreneurship and new job creation and new benefits to our citizens that we serve. Key amongst the challenges is the review and approval by regulators. This has proven to be a very large hurdle in some countries, and it's taken many, many years for regulators to understand the science and the benefits and to weigh the benefits of a technology in the face of a consumer society that is not sure whether they want to accept or to reject. The combination of these two has made it very difficult. What is our job as academicians? In order to move something like this forward, after acceptance by the consumers, of course, then commercial development and successful marketing can lead to the availability of that product. But often the commercial development is very slow to take on new technologies. Whom does a consumer trust? Because clearly the consumer is the person that helps to make that decision of acceptance or not. How do you as a scientist earn trust? Because if you have trust, you have social license, right? Without the trust of the consumer, you don't have the license to do your work and release your work to them. That social license gives you freedom to operate. In other words, to do your work and to develop the products. But if we look at these two, at this balance on the fulcrum, most important is our shared values, not your skill, not how good you are, not how much you know, not what technology you use, but whether or not you have their value. If you share their value, then you have freedom. Shared values are 3 to 5 times more important in building trust than is demonstrating competence. It's very sobering, isn't it? It's not what we know, but what we know about the consumer. That's important. And because what makes sense for research and business does not always make sense for the consumer. So having their trust gives us license. That license gives us power in the regulatory system. And then it's up to the government to make those changes. This paper was published in just several weeks ago, in September of this year, in this volume, in Science magazine. The authors are the same group, many of them are the same group, that wrote a large document published by the National Research Council of the National Academy of Sciences that talked about more tests and the importance of the testing for regulation. This paper is a real change because it says we have learned over the course of the last 20 or 25 years that the technology itself is not dangerous. What's more important is the quality of the product. Is the product, a good fit for agriculture? Does it work? Will the products sell? So the current process-based approach to regulation is no longer, as they say, fit for a purpose and should be changed. I believe have been my opinion for many years that this is an important kind of statement to make. You have seen some of my other some publications that I've had in the past about regulations. And I think science will lead the way, not only for technology, but for regulation, if we know how to explain it to the regulators and also to the consumers. There's a critical role, therefore, for academies of science, as well as the academicians themselves around the world to ensure the well-being and future of societies. We need a trained research workforce at all levels. And in the early remarks of this meeting, we were reminded of the challenges of workforce, and that workforce is important at all levels. STEM technical work, what we call in the U.S. a STEM technical workforce, undergraduates, graduate students. But these should be visions for systems and for collaborative science and technology, with a knowledge of the goals that we as society require for our survival and for the survival of our planet. We need to seek to prepare scientists to become leaders and innovators, not simply to be specialists, but to become leaders and to have an innovative spirit. We should seek to ensure that scientists and STEM workers are good communicators. Communication is learned. It's a skill that's teachable. It's a skill that's learnable. And we need to ensure that our scientists are not only bright and leaders and innovators, but are good communicators as well. And we need to foster relationships with governments and authorities. And then we need to create mechanisms for funding and rewards for innovators that focus on benefiting consumers and the taxpayer. How we do this on top of all the work we do as scientists can be difficult to think about, but you are the leaders of science in your nation. It is your responsibility as much as anyone else. If we fail to take this on, society will be not advancing as rapidly. In the US, we have more than 20% of the population who refuse to take new vaccines. I don't know the number in Korea, but in the US, it's really remarkable to me that the public is not engaged in the protection and the application of science that we as scientists would approve of. So we have lots to learn, and how to be agents for change is what your job is, what my job is. This is not new to you. And I said in the beginning of my talk that much of what I was saying was meant just to stimulate discussion and to stimulate us to activity to ensure that the science and technology, some of which will be discussed by the later speakers, Dr. Van Emmeman and Dr. Chang and Dr. Noboru Noguchi. By the way, I've met you, Dr. Noguchi, earlier at Hokkaido, I think. And Dr. Chung. Thank you all for being part of this symposium. Again, I thank you for the invitation and for your attention. If you have comments, please share them by email or otherwise. And at this point, I will turn the microphone back to the chair of the session. Thank you again for your attention and the invitation to be here today.